Hello everyone, this is Kritika. So today we'll be continuing with the structure of DNA. So we completed till structure of DNA, double helix model of DNA. So now we'll be starting with the forms of DNA. So there are different forms of DNA. So we have bDNA, which is mainly the right-handed form of DNA. Then we have aDNA, zDNA. There are different forms of DNA. So first of all, the DNA, we know that it is located in the nucleus of the cell. So mainly it is located in the nucleus where chromosomes are there. Inside the nucleus, you can find chromosome. Inside the chromosome, the chromosome basically, the DNA is what that completely makes up the chromosome. So the chromosome only it is passed on from the parents to the offsprings and all. So then we have two type of DNA basically, nuclear DNA and mitochondrial DNA. Nuclear DNA is biparental. That means you can get nuclear DNA from both your mother as well as from your father. Whereas mitochondrial is also called as maternal DNA. It is mainly passed on from the mother to the offspring only. And both of these type of DNAs, multiple copies per cell can be produced. Okay. In case of degraded samples, degraded biological samples, especially in hair and all, it is a very good source for the follicular tag of hair and all is a very good source for the DNA, especially maternal or mitochondrial DNA. So different forms of DNA are helpful in extracting from the biological evidences. So then we have polymorphism in DNA. Poly means many, morphism means there are different morphological forms in DNA. So we know that DNA is a double helical structure. It is mainly made up of four different nitrogenous bases, mainly purines and pyrimidines. So purines are the adenine and thymine, and guanine and cytosine are the pyrimidines. We covered that. So the bases, there will be slight deviation or variation in the bases. That only makes the differences in the DNA structure. That is basically DNA polymorphism. As you can see, variation in the DNA sequence among the individuals of same species at a particular locus. Locus is nothing but the location. At a particular location of the DNA sequence, there will be a slight variation in the base pairs. Nitrogenous base pair or the nucleotide base is basically the DNA polymorphism. Clear? And this variation is what it forms the basis of DNA profiling or DNA fingerprinting, basically to identify. So there, see, no two individuals will have the exact same DNA sequence, except in case of identical twins. Identical twins alone, they will be having the exact DNA sequence. Others, everyone at least one or two nitrogenous base pair deviation or differences will be there. Okay, so that is about the forms of DNA and polymorphism in DNA. So this is the structure of DNA which was given by Watson and Crick in the year 1952. You can see the double helical form of DNA. There are four nitrogenous bases: adenine, thymine, guanine, and cytosine. So adenine and thymine are your purines. Guanine and cytosine are your pyrimidines. So there are two hydrogenous bases between nitrogen bonds between adenine and thymine, and between guanine and cytosine there are three hydrogen bonds present between them. And we know that the complete, backbone of your DNA is made up of sugar and phosphate only, as you can see here. That is what is written here. Okay. So now coming to the type of polymorphism, this is one of the very important topic in forensic. So type of polymorphism used in forensics. So we have RFLP, VNTR and STR is there. RFLP is restriction fragment length polymorphism. So as you can see, fragment length polymorphism means there is a small variation in the length of the DNA fragments. See, restriction fragment length polymorphism means you have a DNA sequence. By using restriction enzymes, we are cutting the DNA sequences, and that DNA sequences are then analyzed by using PCR technique and all basically, or by gel electrophoresis also they can be separated. Okay, so that is about RFLP. Now coming to VNTR means, so you're having a homologous DNA sequence and you're able to cut the DNA sequences by using restriction enzymes, and then you're seeing the variations between them, and by either gel electrophoresis or southern blotting you're able to segregate the the bases, and you're able to analyze them. That is basically RFLP. There is a deviation or variation in the length of the DNA sequence. Now coming to VNTR, variable number of tandem repeats. As you can see, repetition of the fragment, repetition of the base pairs is basically there. And the repeating sequence is between 15 to 100 base pair units are there. So the base pairs are repeating after 15 to 100 base pair. The base pairs are kept on repeating. Okay, so VNTR basically you can see means from the head to the tail region, it will be taken into consideration of the DNA sequence, and the sequence will be keeping on, the repetition of the sequence will be there. That is called as variable number of tandem repeats. Number of tandem repeats are more, and there is a variation after the tandem repeats. Now coming to STR means that is short tandem repeats or microsatellites. So these are actually called as the gold standards in forensic science, especially in case of DNA profiling. Why? Because there is only short repeating units are there. You are taking only 2 to 6 base pairs in consideration, and after 2 to 6 base pairs, in that count, there is repetition of the base pairs is happening again and again. So this is a very good so to say polymorphism form of DNA which is used in forensics and all. So for example, CODIS. CODIS is basically combined in database in combined DNA index database system. So basically where all the DNA bases will be collected and will be put into that database particularly. So for DNA profiling and all, this particular STRs can be really helpful for us. So now coming to polymorphism. Polymorphism, as you can see here, it is the occurrence of the alleles. So here you can see it. I'm sorry. Here you can see, right? Sorry. Polymorphism. So here there is a variation in the DNA sequences. So we have structural polymorphism means the length of the DNA sequence, there is a variation. Copy number polymorphism means number of copies of the DNA segment or fragment that has been generated. There is a variation in the number of copies of DNA fragment that is produced. That is called as copy number polymorphism. So these are the two different type of DNA polymorphism. So mainly to study about genetic diversity, to study about disease and association with them, for evolution, for population genetics and all, polymorphism is highly helpful for us. Okay, so when you're coming to the type of DNA polymorphism, we have SNPs, that is single nucleotide polymorphism. From the name itself, you can understand only one particular nucleotide, there will be a variation there. Okay, only one nitrogenous base or one particular nucleotide alone, there will be a variation there. It alone differs. So when you take one complete DNA sequence, mean one particular nucleotide base alone, there will be a certain variation. Okay, there will be some difference in them. That is called a single nucleotide polymorphism. Now, insertion or deletion polymorphism means either one nucleotide base pair is either inserted into the DNA sequence or it has been removed from the DNA sequence. That is insertion or deletion polymorphism. Either they are inserted or their one particular nucleotide base is getting deleted from that particular sequence. Now we have polymorphic repetitive sequence as well, where I have microsatellites, macrosatellites, and mini satellites and all. Okay, and then we have structural and copy number variation. As I told, a number of copy of DNA fragments that has been generated, there is a deviation in them. These are the different type of DNA polymorphism. Okay. Now, coming to SNPs and mini satellites. Single nucleotide polymorphism means, as I told you, at a single base position alone, there is a variation. In case of degraded samples and for phenotypic prediction and all, they are being very much useful for us. Phenotype is mainly the physical characteristics here. Genotype is the genetic characteristics of an individual or an organism. Phenotype is more on the physical characteristic features like skin color, hair color, those kind of things and all. Okay, so to analyze about them in detail, SNPs are highly helpful for us. Then we have mini satellite. Mini satellite are nothing but the tandem repeats. I told you, 10 to 60 base pairs alone, there will be a tandem repetition of the base pairs will be there. These are used in RFLP based profiling and all. Okay. And now here you can see the significance. These polymorphic regions are non-coding. They do not code for proteins or anything. They are being inherited in a Mendelian fashion alone. So for individual identification, for paternity, maternity analysis, for analysis of degraded, for analysis from the degraded samples, for forensic matching, for every single thing, SNPs and mini satellites are highly helpful for us. Now, coming to RFLPs. So, as I told you, restriction, fragment length, polymorphism, there is a differences in DNA sequence. So, the complete length of the DNA sequence, there is certain variation is there, which will either result in the differences in length of DNA fragments between individuals. So, how do you do RFLP? First, you do DNA extraction. First, you take any biological evidence, whether it be blood, semen, saliva, hair, follicular tag of hair, anything you can take, and you're extracting the DNA from it. Then you're digesting that physical biological evidence, you're digesting with a restriction endonuclease. It can be E. coli as well, that is most commonly used. So by using restriction endonuclease, you're cutting the DNA at specific sequences. Then by using gel electrophoresis, based upon the size and charge, they are being separated. So on agarose gel electrophoresis, where agarose gel is there, and then based upon the size and charge of the sequence of the basically mol enzymes and all, they are being segregated or separated. The fragment is separated in the gel electrophoresis. Or we can do southern blotting as well. So the DNA is basically transferred from the gel, okay, onto a nylon or intercellulose or nitrocellulose membrane. So DNA is extracted, digested with restriction endonuclease, then it has undergone gel electrophoresis. After that, it has undergone southern blotting. So this is how we do RFLP procedure. Okay, so at last we can do the hybridization. We are having any radioactive or labeled isotopes like sulfur, phosphorus, any radioactive isotopes you are taking, okay, which is specific to the VNTR region, and then we can see beautiful bands will be produced there. So that bands are nothing but indicating the DNA fingerprint. Okay, so this is a very important topic about RFLP. The procedure is very important. Clear? And the next very important topic is PCR. So PCR is mainly used for amplification of DNA sequence. First of all, what do you mean by amplification? Amplification is nothing but producing millions of copies of DNA sequences are produced, means that particular process is called as PCR, polymerase chain reaction method. Okay, so what are the steps involved here? So for each cycle, they're selling one cycle, they're telling the steps. So we have to do 25 to 35 cycles are repeated. So in one cycle, first step is denaturation. Denaturation is basically like degradation only. Okay, so we are passing a lot of heat at 94 to 96 degrees Celsius. The double stranded DNA helix will now get separated into two single strands. And then it will undergo something called as annealing. Annealing is where the DNA primers will bind to the single stranded DNA sequences. So for example, let me draw and show to you. So this is your double stranded, it's a very flat diagram. So, okay, now when you're doing denaturation, it has been divided into two strands basically. And I'm adding short DNA primers. So primers will go and attach itself to the single DNA strands. Okay. And the annealing temperature is 50 to 65 degrees Celsius. Then the last we are doing is elongation. What is elongation? Means Taq polymerase or DNA polymerase. We are adding to the two generated complementary strands by using, nucleotides are basically synthesizing two new complementary strands are being generated. So you're getting a proper now newly synthesized elongated DNA strands are being now generated. So denaturation, annealing, elongation. So at last, what is happening? Amplification is done. Millions of copies of one particular sequence of DNA has been produced by the process of PCR. Clear? Now, after amplification, what are we doing? They can be analyzed via capillary electrophoresis. Then you're adding again the fluorescent labeled dyes you're adding. And now you can get the DNA profiling or DNA fingerprinting region can be observed clearly. Similar to the RFLP only. But PCR is a more, it is a more accurate method, a more sensitive method as well. Because even for COVID profiling and all, we use RT-PCR, right? RT-PCR is different, where basically DNA to RNA, when it gets converted, we call it as transcription. RT-PCR is where COVID has the RNA as a genetic material. So RNA to, it basically gets converted into DNA. That is reverse transcription reaction. So for this reverse transcription reaction for COVID testing and all, we use something called as RT-PCR. Clear? So that is about PCR and amplification. So you can see the merits and demerits of the both RFLP and PCR. Merits is that they have high discriminating power. Only specific patterns can be observed very detailed, and it's a well established, a sensitive and a legally accepted method as well. Demerits is that it's a very expensive, a time consuming method, and it uses radioactive isotopes and all, which is labor intensive and expensive, and it requires large quantity of high quality sample is needed. These are the demerits. Okay. Same thing, whatever I said only, it's repeated here. They require, but the merits, so here in case of RFLP and PCR, RFLP, the process is that it's a high discriminating power, it's well established and all, and the demerits are these things. PCR is that you can need a very less sample also is fine. Degraded, old samples, everything, it can give accurate, proper results, a highly sensitive and a specific method. It is automable and cost effective as well. For modern STR and for DNA profiling, for CORDIS databases and all, we are using PCR only. Demerits is that any small contamination, even for the person who's like sending the specimens into the PCR machine also, even slight contamination from them, any foreign particle goes means it can give false results. Okay, mutation and errors can happen because that will again during amplification there will be possibility of mutation and errors are there. Mixed samples also it's very difficult to interpret. So it's a highly sensitive method. So we have to be very careful in handling it. Okay, so now what are all the advanced methods of DNA examinations? We have STR. I told you it's a current gold standard method. Okay, multiplex PCR amplifies a multiple STR loci simultaneously and they are analyzed via the capillary electrophoresis. Mitochondrial is a maternal DNA, especially in your teeth, bones, hair shaft without root and all for maternal lineage tracing, for maternity testing and all. This particular method is highly helpful for us. Okay. Now the next is why STR analysis. So why STR as a name itself suggests, so we know that females have XX chromosome and males have X and Y chromosome is there. This Y chromosome is the one which is passed from the father to the child. This Y chromosome is the one that determines the sex of the child. We know that, right? So if the X chromosome from mother and X chromosome from father comes, means the child will be of a girl child, XX chromosome. If one X chromosome of mother and one Y chromosome of father comes, means the child will be of a boy child. So Y chromosome is a paternal. So from grandfather to father, from father to the child, and from that child to his child. So it goes on. So Y chromosome is a very important case in paternity analysis, in sexual assault, rape cases, and all. It is very, very important for okay. It is a very important tool. It's useful as well. SNPs, as I told you, single nucleotide. As only one nucleotide base, there is a deviation or variation. For severely degraded samples and all, for analysis of phenotype, especially eye color, hair color, and all, SNPs are highly important. Then we have something called as next generation sequencing. So here, next generation or massively parallel sequencing, a multiple number of what to say, simultaneously you can analyze DNA sequences in very detail, and it helps us to give a proper, accurate, and high resolution detail or results can also be obtained from them. That is about next generation sequencing. Okay. So now, DNA phenotypic or forensic DNA phenotypic. So as I told you, predicting any kind of physical traits. Okay. And there is no DNA database to match everything and all. So to predict the physical traits and all, where basically we are doing forensic DNA phenotyping is done. And touch DNA is wherever the places you touch, your DNA is basically transferred to that particular place. So when you do DNA analysis or testing, when we have recovery kits and all, no, you can easily analyze the touch DNA from it. Then we have rapid DNA technology is there. It's rapid, it's very fast. Within like 1 to 2 hours itself, the results are generated. Okay, on site, you can easily produce the results. So you can see RFLP, PCR, and YSTR analysis. This is a particular method. See here you can see you have different samples I've taken. They have run on gel electrophoresis, then they are taken to southern blotting. Bands have been obtained for different samples. PCR, as you can see, they have done denaturation, annealing, and elongation extension is basically done. YSTR, as you can see, this Y chromosome analysis can be done to analyze any kind of paternal lineage testing or for sexual assault case or for rape cases. This is basically used. Okay. Then we have something called as DNA methylation analysis. So basically what happens is that to your DNA sequence, a methyl group, that is CH3 group, is basically attached to this. So there are certain epigenetic markers for each and every body fluid that is obtained from a crime scene. There are different molecular markers that has been available to identify the age, that is basically how old is that particular biological evidence. Is it old or is it new? To analyze that, there are different epigenetic markers that is available for distinguishing between blood, saliva, urine, semen, feces, and everything. Different markers are there. Okay, and the technique is basically DNA methylation analysis. Then we have CODIS, Combined DNA Index System. It's the national DNA database where all the DNA profiles are being of the offenders and everything are being stored there. So for easy analysis and for comparison and all, we have this CODIS. Okay, so that is about it, guys. Thank you.